Good morning. It's a real pleasure to speak once again in this prestigious symposium, this time to honor my good friend and collaborator Jerry Tuscan, a real icon of forest genomics. And of course, it's also a pleasure to be with so many other good friends today. Too bad I could not be in person this time. Going from tree genomes to future forest, I'll briefly try to show how the new science of genomics and the old science of quantitative genetics have converged to cause a paradigm shift in tree breeding. Genomic selection is becoming the applied decoding of the genome, taking us to the modern breeding of future forests. I met Jerry in 1991 at the famous Gatlinburg Symposium on marker-assisted selection. It was a landmark event that started this long journey toward the use of genomic data to impact breeding. It was in 2004, however, that we started working together on the Eucalyptus genome sequencing project under the lead of Zander Meyerberg. This is when we took this historical picture in front of a giant eucalypt in Tasmania. So, Jerry, congratulations. Thank you very much for all your contribution to forest genomics, especially, of course, the sequencing of the Eucalyptus genome. The challenges we face for planted forests at all levels, industrial or small farming, are significant. Much higher demand for wood is predicted, but with less and less land available or viable for plantation due to global climate change, particularly regarding changing rainfall regimens. This is particularly important in the case of eucalypts. Now, as we have limited agency to significantly alter the environment, the challenge is faced through genetics, genomics, and breeding when developing more adapted material. So basically, we're trying to understand how genetics controls the variation in traits like tree growth, stem form, wood properties, and stress tolerance, such as drought, for example. Tree breeding has some specific features and hurdles when compared to crop breeding, and some are particularly relevant. Almost all traits of interest are complex, except for some chemical properties, such as lignin, where important advances have been made with the manipulation of single genes. Breeding cycles are long, complex and long-lasting interactions take place between genes and the environment during the long lifetime of trees. And usually we require large areas and long-term investments, which are more susceptible to changes in market demands and the realities of changing climate. Tree breeding has evolved quite a bit in the last 30 years. We have well-developed methods of flower induction, for example, to allow going from seed to seed in 1 year in the case of eucalypts. We can generate large full seed families, clonally propagate almost all genotypes, and used advanced field trial designs for measuring traits of interest with very good accuracy. And in the case of eucalypts, we can also access the wide interspecific variation to efficiently exploit it in hybrid combination to produce real genetic innovation. In this example, by crossing Eucalyptus globulus with the frost-tolerant Eucalyptus nitens, we produced frost-tolerant trees. However, field testing and selection still require several years, even in fast-growing eucalypts, between the time we generate the cross, going through all the steps, to the time that we actually recommend a clone for commercial plantation. So any indirect, early DNA-based selection method or direct gene manipulation would be a game changer to impact target traits and accelerate breeding cycles. This is where genomics comes in. This, however, depends on our ability to map how the variation in genes and genomes directly contributes to the observed phenotypic variation. The question posed here is, of course, what every geneticist is trying to answer in all organisms, and two approaches have been taken to this end. A more Mendelian approach, based on the idea of finding and manipulating single genes of large effect, and a quantitative one, polygenic, based on the idea that many genes control complex traits. Now, this debate, of course, was resolved many years ago by Ronald Fisher, infinitesimal model of many genes of small effect controlling complex traits. The advent of genomic technologies has profoundly impacted these two approaches by allowing, on one side, faster gene discovery, and on the other side, higher predictive power when genomic data is associated with the quantitative genetic methods. Many gene discovery studies have been published in mainstream forestries by genome-wide association studies, where we have tried to associate DNA markers to complex traits and possibly find the underlying genes. Jerry's team, ourselves, and others made many contributions in this area. However, results have not been promising from the applied standpoint. Traits relevant to productivity are polygenic. Hundreds or possibly thousands of genes and their interactions are involved. Detection of this gene is very challenging unless huge sample sizes are studied. Effect sizes are small, usually less than 1% of the genetic variation, and frequently not well estimated, and their validation is challenging. So from the applied breeding standpoint, we do not expect significant impact for complex productivity traits in the near term. Now, we should not feel alone about this. This has been the rule in the top GWAS studies for human height, for example, a trait with strong genetic control. Now, in this study, more than 5 million individuals were studied. 12,000 gene trait associations were detected, covering 21% of the human genome. So 21% of the human genome participates in the control of human height. But only half of the genetic variation could be accounted for by those 12,000 associations. The rest remain undetectable. So this is probably what we would find for tree height and drought tolerance, for example, if we were able to carry out such a huge experiment in a forest tree. And independent validation of the associations found has been very challenging for many traits and diseases, even in humans, where many studies have been done. Validation rates are usually less than 10%. In trees and plants in general, there is very little independent validation. Jerry's team has made the largest advances in this area in forest trees. Recognizing that the architecture of complex traits is highly polygenic, the convergence of the new genomic technology and time-proven quantitative genetics has now resulted in this new paradigm. For applied breeding, we have moved from trying to discover individual gene to capturing the full aggregate of the genome effect like quantitative genetics has historically done, but now using genomic data. Taking us to the very concept and practice of genomic prediction of complex traits, a field that has experienced a huge development in the last 20 years in animal, crop, and tree breeding. Now, genomic selection follows a very simple concept. You select on a large number of markers to capture the entire genome-wide effect in predictive model, irrespective of understanding the underlying mechanism. So imagine a genome with 5 chromosomes, these black bars, and you have an unknown number of genes, the red squares, of unknown effect. You just use a large number of markers, these gray arrows, to select on, and therefore capturing the effect of all those genes that are controlling the traits of interest. Yes, of course, it's a black box method. I call it the black box of how evolution created all those mechanisms. Now, the practice is also relatively simple. Mathematical models are trained based on field data, here in the parental generation, where trees are phenotyped and genotyped in what we call the training population. Now, this model is then validated in an independent validation population, and once we have a good, accurate predictive model, we can use it to select on the seedlings derived from that generation at early age and carry out what we call the genomic selection cycle. So, these genomically selected seedlings are then ranked based on their genomic predicted genetic value, and depending on the interest, you can either clone them and test them in the field for validation, or you can induce flowering quickly and cross them and turn a new breeding cycle in the program. This approach, of course, can radically increase genetic gain per unit time, which is the measure of efficiency of a breeding program. We can increase selection intensity, which is the number of individuals that we can screen. And of course, the more individuals we can screen, the better chances are that we find the best ones. We can increase selection accuracy, which is the correlation between the genomic breeding value and the true breeding value, because we can get closer to the genetics of the individual. And we can reduce breeding cycle length. So by tinkering with those three variables, we can increase genetic gain per unit time, increasing the efficiency of a breeding program. To carry out genomic selection, we use SNP markers. Single nucleotide polymorphism is the most common variation in genomes. For Eucalyptus, we have developed operational SNP platforms used worldwide for the 20 or so Eucalyptus species by more than 50 companies or research institutions. Now, we currently genotype at the cost of about $25 per sample. Now, genomic prediction in trees typically show predictive abilities between 50 up to 90%, depending on the trait and the population. In the y-axis here, we have the observed breeding value, and the x-axis, the genomically predicted breeding value, for 15 different traits in Eucalyptus. Chemical traits such as lignin content and composition, or cellulose, have higher predictive ability because they are relatively simpler traits, while growth and physical fiber traits usually in the range of 50 up to 70%, depending on many other variables. In this particular study, that took 7 years to complete, we showed for the first time prospective genomic selection results across generation in a forest tree. We reached high prediction ability and, more importantly, high ranking correlation between what trees we would have selected at harvest age at 6 years, on the left-hand side here, the ranking, compared to what we had genomically selected at age 3 weeks in the nursery on the right-hand side of the graph. So we could get a ranking correlation of 87%, and within the 5% top selected individuals, we could correctly rank 60% of them. Again, we're not alone. In the last years, human geneticists have adopted the same genome-wide prediction approach of complex traits with the so-called polygenic risk score, an estimate of susceptibility to a complex trait or disease. For example, in this particular study, 2.1 million DNA markers were fitted in a model to predict obesity. The accuracy reached here was about 40% in the top decile. So we actually get better accuracies in trees and plants in general, as we have seen before, for a number of reasons that I won't have time to discuss today. But the interesting point here is that in human genetics, the same approach is being used not to select, of course, but to predict the risk. As a genomic breeder, I've used an analogy, which is also a bit of a tease with my fellow molecular biologists. Genomic selection is like investing in index funds. It's cheaper, simpler, humbler, and generally more efficient. Instead of trying to pick single winning stocks or genes, we breeders simply buy the whole market, or better said, the whole genome effect. Now, many financial studies show that this tends to be more efficient on the medium to long term, and we believe this is the same case in tree breeding. In conclusion, these are the advantages and remaining challenges of using genomic selection in tree breeding. We can increase genetic gain by accelerating breeding cycles and especially increasing selection intensity. However, the environment continues to be a moving target, so that predictive models have to be updated regularly with new field data. Cost of genotyping is still an issue, but it's coming down. And of course, a criticism can be made that we're not advancing in any mechanistic gene discovery and understanding, if that's the objective. To deal with the challenge the environment represents, environmics approaches have seen a rapid development in recent years. Just like genomic data allows measuring and predicting the relationship between tested and untested individual trees, environmic data, which is large-scale environmental data, allows doing the same between tested and untested environment. In Brazil, we have done a significant progress in using large-scale environmental and genomic data together to guide the prediction of performance of untested trees in untested environments. Research and implementation of genomic selection programs in tree breeding has been internalized by several Eucalyptus forest-based companies worldwide, using the genomic platforms we have built and developed and the approaches we have shown. So a large number of publications have been reported in recent years, and we see a very bright future for the operational use of this technology. I'd like to thank all the collaborators that worked with me on this journey since 2008, actually, when we started working on genomic selection. And thank you very much to all of you for your kind attention today.
Dario, thank you very much. I was going to say, I can see Dario, but you can see him too. It's great to see you. You look fantastic. Just very sorry, Dario, that you're not with us.
It's okay. It's, next time.
Next time. Next year, you promise us. I've collected a few questions from some of the team here, some of the young people, and I'll throw those at you because you've given us some time to deal with questions. I'm not going to ask for questions from the floor because that'll take too long, although I am tempted. But Dario, I thought you've listened to the symposium through, and what time is it there? It must like 5 a.m. or something.
Oh, it's 7:20.
Oh no, I was feeling sorry for you. Yesterday we were speaking about poor old Dario, but coffee is very important in Brazil, so you you kind of keep yourself awake. But yeah, cheers. Dario, you've listened to the presentations thus far. I was wondering whether you don't have a comment or two to make, perhaps to Jerry or to Steve, well, I have.
Jerry and Steve have been my mentors in many ways. So I'm just so happy to be able to have worked with Jerry and also met with Steve and so I mean it's just great talks, great stories, and the glass sharing was also funny. So
yeah, I'm still thinking about those bacteria, but we won't go into that now. So Dario, genomic selection. I mean, you've done tremendous work, you and others, but you're really the leader in terms of eucalyptus plantation forestry. Listening to, for instance, Jerry. So there is a dichotomy, the whole haystack or the gene. What do you feel about that? I want to pivot that particular question towards, for instance, regulation and genetic modification. And CJ's talk that follows, and you know her as well, will get into the genetic modification space. But how do you feel about that?
Well, actually, they are extremely complementary. I mean, I think there is a great future for both approaches, especially when they're combined. And I'm sure that the concept of identifying and manipulating single genes of larger effect will become more and more useful in breeding, although the challenges are definitely not small for a number of reasons that I mentioned some of them, but they are totally complementary, especially when we have traits for which we do not have enough variation in our gene pool in the specific species we're working with. So, I mean, for example, in lignin manipulation, there have been just great advances, and I'm sure that this is an area that the prospect of CRISPR gene editing, multiplex gene editing, has been shown actually, and so I think it's extremely useful and complementary to the way that we're looking at polygenic traits related to productivity and adaptability. So that's my short answer
So
for that.
that was a piece of a question that I had on the paper. But you know, there's this whole issue of regulation, and you heard the presentation that we had from Jessica, so European Parliament, relating to regulation. I mean, this is a big issue for us, is acceptability of, say, for instance, genetically modified or genetically edited trees. How do you feel about that? I know Brazil is quite far ahead in that particular space, but integrating that into intensive plantation forestry using, for instance, eucalyptus, so a non-native species in a naive environment, I guess.
Yeah. Well, I think, Steve Strauss, he's the expert on this topic. So, it's very difficult to say anything. So I think, again, especially in eucalyptus where we can clonally propagate relatively easily, I think the prospect of being able to use genetic modification, especially now with the gene editing technology, I think are great. Again, the challenge is really having the good target, understanding the mechanism, how to edit whatever we want to edit. And I think the future, as Jerry put it very clearly, I think the future for the next 20, 30, or 50 years are bright. And I have no doubt that acceptance will happen, especially as we face increasing challenges in terms of being able to plant and grow trees in different environments. Here in the tropics, we have upcoming challenges, especially related to water distribution or rain distribution. And so I'm sure that the combination of a more polygenic view of adaptability trait together with traits where we can generate new mutants, let's put it this way, new phenotypes that we're not able to generate by conventional breeding, will be key for the future of our planted forests.
Yeah, you talk about the challenges, and as you know, I'm particularly interested in what pests and pathogens are doing. In fact, I think maybe water, but I think that is the single greatest threat to plantation forestry globally. A subject for another time, but I will tell you that I was very interested in one of your slides, emasculating eucalyptus flowers. And you have a really interesting disease on those leaves. So I'm wondering whether you go look at your slide. I am wondering whether you're choosing the best trees, but that also for another time. You've disappeared from my screen, but I can see you at the back. So Dario, you made an interesting point, and Jerry did too, that in many ways the technologies that we utilize and that are being utilized in tree breeding and genomics relating to trees has followed human genetics and human studies. And you gave examples of that. You sort of said, but we're not so bad and we're more or less better than them. But I mean, a question in my mind, for instance, and it also was captured in a point that somebody made in our discussions here, was what are the medical researchers? Firstly, why are the medical researchers ahead of us? Where are they now? What are they doing that we're not doing that we can learn from? And maybe the other way around, what should they be learning from us?
Well, I think, funding, it's very different in many ways. And the buildup of huge databases, like, for example, the UK database where they just resequenced several million individuals and they have phenotypic information on clinical data for the last 50 years or sometimes even 100 years. Sweden also has also done some huge developments in this area. So I think we really would need something similar, because we learned that especially for dealing with complex traits, we need huge sample sizes. And of course, Jerry has done some of the best efforts in this area with poplar. He mentioned the resequencing of 1,500 trees, but we'll probably need more like 15,000 or 1.5 million trees resequenced and phenotyped in the field, different environments. And that's something that we might be able to do in the future.
And you made a very, very important point, maybe a sensitive one to all of us, and that is the issue of money. You know, I oftentimes think about the COVID pandemic, and my physician said there's no vaccine coming, and the reasons. I mean, he showed me Fauci's book, and a year later we had vaccines, and many of us might not have been here today if that hadn't been there. So what happened there, other than the so-called blue sky fundamental research that was done many years before and probably got a lot of criticism, what really happened is they threw the bank at the problem, the money. So yeah, I guess we've just got to keep pushing for the funding, keep convincing the industries, yeah, that there's something to be gained. But it did make me think about, you've done this tremendous work on eucalyptus, and I mean, these huge areas. I think I looked the other day, about 35 million hectares of eucalyptus planted in plantations. That's non-native eucalyptus planted in plantations. But you know, there are other trees. So what about acacia, for instance? And there are many, many millions of hectares, not the same as eucalyptus, but you know, approaching that. Why are we not seeing the same drive for eucalyptus pine? I mean, for acacia, pines a little bit more complicated. But I'd like your thoughts on that. And then we're going to stop.
Well, actually, in acacia, there have been great efforts in Indonesia. I've followed that, including some publications, and they are already applying genomic selection as well. They just don't publish a lot because those are companies investing, so that they're not, you know, driven to publication. Of course, in conifers, great efforts, great advances have been made in the US, in Canada, in Sweden, and you know, and Harry Wu will talk today, so I'm sure that he will tell us great stories. So I think overall we see a great interest and moving forward into the use of genomic prediction, genomic selection, and breeding program.
Yeah, Dario, we have to end. I hope you stay with us. We see you on a big screen. You look great. No, really, and the presentation worked really well. When people say recorded and online, I think, oh, here we go. But we have fantastic technologists here, so we must be grateful to them too. Thank you so much for joining us. Enjoy your coffee, and we continue.
Thank you very much.
Take care. Bye.
Thank you very much, all of you.